What's a gene? A gene is a section of DNA that codes for proteins. In this video, we're going to learn more about genes. We're going to learn about how much DNA is in the form of genes and why it is that different people have different forms of the same genes. You may be surprised to learn that there's such a thing as dogma in science, but a long time ago, several leaders in the field of molecular biology, which was then a fairly new field, came up with what they called the central dogma of molecular biology. And that central dogma, which is illustrated here, is that DNA, in the form of genes, produces RNA through a process called transcription, and that RNA then produces proteins in a process called translation. Now, this central dogma, this process of DNA to RNA to protein, is very interesting because you'll notice that what it involves is such things as sending messages. The DNA produces a type of RNA called messenger RNA, and something called translation. Where are the genes located? Well, the answer is in the nucleus of cells for vertebrates, animals, plants, and in bacteria, the DNA, the genes are on DNA just free-floating within the general cytoplasm of the cell. In advanced vertebrates, mammals, etc., the DNA is organized into features called chromosomes. And if you look at the picture of DNA here, you'll see that the word gene does not cover the entire section of DNA, but only part of it. And that's because not every part of the DNA of a cell are actually genes. Let's take a look at this in some detail. The total DNA in a cell is called its genome, and the genome is not made entirely of genes. In fact, genes make up about 25% of the total genome, and the rest of the DNA are not genes. Even in genes themselves, the vast majority of DNA does not code for protein. It's in fact non-coding. It turns out only 1.5% of the entire genome actually codes for proteins, does the work of the gene. If we look at a section of DNA, this you may be familiar to you from the previous video, DNA 1, that series of bases of nucleotides, which as you know are organized into 3 base codons, we can look at that in a larger stretch of DNA, and you'll see it occupies a small black box. Now, there could be several. In this case, we have 4 of those boxes of different sizes, different lengths, separated by gaps, also of different lengths, and the sum of those 4 boxes are called a gene. The boxes themselves are called exons. And those are the parts of the gene that actually code for the protein. The areas in between, where there's no box, are called introns, and they have no role in the coding of proteins, as far as we know. Now, in case you think that astronomers are the only scientists who deal with very large numbers, we biologists do as well, especially in molecular biology. For example, in a single human cell, there are 3 billion base pairs. In other words, 2 sets in a double helix of 3 billion nucleotides, or bases. The number of genes in a human cell turns out to be 20,000, which was a big surprise when it was discovered because many plants and animals have far more genes than we do, including the rice plant and a number of animals. It turns out the number of genes is not terribly significant when it comes to determining the complexity of an organism. And those 20,000 genes take up about 750 million base pairs. It's a pretty big number. And that, remember, all of these are in one cell, not in the entire organism. And again, that's 25% of the total amount of DNA in the cell. Within the genes, the 750 million base pairs, 46 million base pairs are in the exons that actually code for proteins. So the total percentage of the base pairs in a genome that actually code for protein is between 1 and 2%. Now, that really doesn't make much sense. Here we have DNA, which is a huge molecule in a cell, and only a tiny fraction of it, 1.5%, actually does what we think DNA is supposed to do, namely code for proteins. This picture is a drawing showing roughly what it looks like if you looked at a section of DNA in a chromosome. There might be 2 genes there. If you look at gene 1, you see 5 exons. Some are skinny, some are a little fatter, separated by various lengths of absolutely non-coding DNA. Those are the introns. Gene 2 has 6 exons, and there are genes in various animals with hundreds of exons. Some have only a few. And we don't really know what the introns do. But what's very surprising about this structure, and was very surprising when it was first discovered, is that when the gene produces a messenger RNA with all its information, both introns and exons are copied, and the introns have to be cut out. It seems like a huge waste of effort, and it's something that is still not fully understood. So all of this raises the question, if only 1.5% of our DNA in the entire genome actually codes for proteins, what does the rest of it do? That's 98.5%. The great majority of DNA does what exactly? Hopefully it won't come as a surprise to you, although it might, that science is just as full of controversy as anything else, like politics, religion come to mind. Usually those controversies eventually get resolved by more and more data. But there's always a time during which the controversy is boiling because different people have very different views looking at the same data at the same time. In this case, we're going to talk about a controversy related to junk DNA. What does that mean? Well, when it was discovered that so little of the DNA actually belongs to coding sequences within genes, and it was also discovered that large sections of DNA are clearly not useful. There are thousands of base pairs in a row that are simply AT, AT, AT. We just get what are called repeats that have no possible bearing on the function of genes. So the conclusion was reached that most, if not all, of the DNA that is not part of a gene, and even the DNA within the introns, are simply useless and just are there for various accidental reasons, and it was called junk DNA. Some of those sequences that we call junk DNA are also viruses that infected a genome a long time ago and are now no longer active. There are genes that are no longer active called pseudogenes, and they don't have any value. And there are other kinds of either repetitive or other types of DNA that are probably definitely useless. But we also know there are parts of that DNA that do have some function. Some of them regulate gene expression, some are copied into RNA strands that don't code for proteins but have other functions in gene and enzyme regulation, and some have the capacity to produce new genes. There are sections of DNA called transposons that can change the way genes are expressed or regulated. A few years ago, a project at the National Human Genome Research Institute called ENCODE published a striking and very controversial paper suggesting that 80% of the non-coding DNA actually has a function. There was a lot of pushback against that, many scientists saying that some of their conclusions were poorly drawn from the data, and the amount of functional DNA in the non-coding regions must be much less. So, at this point, we really don't know what the answer is. My guess, frankly, is that we will find that as we get more and more data, and this has already been said by a number of leading scientists in the field, we will find that more and more of what used to be considered junk actually has function. One of the very interesting things that I will be talking about in a future video is the way genes are regulated. And gene regulation turns out to be an extremely important topic and probably explains why we can get away with only 20,000 genes and still be the advanced species that we are. So we are just at the beginning of learning about this issue, and hopefully at some point in the future this issue will be resolved. Now, one thing that's an important part to understand in molecular biology is that all members of a species have the same genes. All elephants have elephant genes, all pineapples have pineapple genes, and all humans have the same genes, human genes. But there's a caveat here. The same genes are not the same for everyone. If they were, we would all be identical. We would be clones. We're not. We're different. And the reason that all individuals among humans or any species are different is because we have small differences in our DNA sequence in the genes and in the parts of the genes that code for proteins. Those differences are called alleles. To understand what an allele is, let's take a human population. As we know, humans are diverse in the way they look and in many other aspects of their biology. It doesn't mean that anybody has different genes from anyone else, but the exact sequence or the exact form of the gene is slightly different. For example, person 1, circled in red, may have for a particular gene a sequence of DNA which we will call allele 1, and you can see its sequence there. Another person, also circled now, has the same gene, but in that particular stretch of DNA there is a change in the sequence. What used to be, what is a G for the first person, is an A for the second person. So that person has allele 2. Another person may have a different allele. Some genes have 2 or 3 alleles, some genes can have 30 or 40 alleles, there are some genes with over 100 alleles. In this case, let's assume that the woman, person 3, has a third allele. The A in allele 1 and 2 has become a C. Now, what if all the alleles were the same, not only in this gene, but in every gene? In other words, let's say you had 3 people who have exactly the same DNA sequence in every gene in their cells. What you see is the picture. These people are clones, in this case, identical triplets, and that is exactly why identical twins, triplets, etc., look exactly the same. Because all the genes that code for things like hair color, size, eye color, etc., are identical, and so that's why they look identical. Let's take a look at this a little more carefully, and this time we're going to look at it next to the genetic code, which you may remember from the first video. The first allele, and now they're broken up into codons, so you can see that ATT is the codon for isoleucine, GGC for glycine, AAC is a codon that means asparagine, ASN, and GTT is the codon that codes for valine. Those are 4 amino acids, and these are 4 of the codons that code for each of them. That was allele 1. Now, if we look at allele 2, we see that the difference between the 2 alleles is that the 5th nucleotide, or the second base in the second codon, the G, has become an A. So if we look at the genetic code, we see that before we had GGC that coded for glycine, and now we have GAC that codes for aspartic acid, and that's circled in green. So the protein sequence, that part of the sequence of the protein, has changed from a glycine to an aspartic acid. What is the effect of that mutation? We don't know just by looking at that, but we will discuss that in the next video when we go into details in mutations. Now, what about the 3rd allele? Here, the AAC codon, which codes for asparagine, has turned into ACC, which codes for threonine, the blue circle. Again, we now have 3 alleles that produce 3 different proteins. Now, there could be another allele with another change. In this case, it's also in the second codon, but what has happened is a C has turned into a T. If we look at the genetic code table, what we see for glycine is that GGC and GGT both code for the same amino acid, glycine. So even though there's a mutation in the DNA, a C became a T, the protein is completely the same. So this is an example, which we'll talk about more in the next video, of how you can have a mutation in the DNA that has absolutely no effect because there's no change in the protein that is produced. Now, with all these changes in sequence, with all these different alleles going on, it might seem that humans are really different, and perhaps even different species or different subspecies. That is not the case. In fact, the genetic diversity between people, despite appearances, is much lower than it is among most other primates, including, for example, chimpanzees. FST is a statistical measurement of genetic diversity. And if we looked at all humans, the average genetic diversity is about 0.05, and for chimps, it's about 3 times higher. Well, what about population groups? This photo shows people of different races. Don't they differ between each other in genetic diversity? And the answer is they do to some extent. This is what's called a phylogenetic map of human beings. And on the very bottom, you see the genetic distance. And what we see here is that the maximum distance is 0.2, which again is low compared to many other species. What you also see is that the branching is continuous. It's not easy to say which of these branches constitute actual races and which are actually part of the same population. I'm not going to discuss this more now, but I will have a whole video on this issue of population diversity in terms of genetic distance to show you that what we call race is actually not a very good biological construct. So this is just about the end of this video, but I do want to bring your attention back to this photograph of the different alleles and the different mutations that occur. In the next video, we're going to look at what is the effect of these mutations. Are all mutations harmful? Are some mutations beneficial? Is that possible? Are there a lot of these neutral mutations, like the 4th one, the 4th allele down, where In fact, nothing changed when the DNA was mutated. How frequent is that? We're going to look at mutations in some detail, but for now, that's the end of this video. Please don't forget to subscribe and like the video. Make as many comments as you like. Again, feedback is still much appreciated. Thank you, and God bless.